Thank you for the introduction. It's a great pleasure to be here and see so many familiar faces. Believe me, I'm very, very happy because of this. And today, yes, I will talk about use of enzymes for the preparation, actually for the preparation of the samples for micropollutants analysis. But what I mean about the micropollutants here is a microplastic. Disposal plastic products became a very popular since the beginning of the production in the middle of 20th century. They are quite cheap, quite easy information, quite durable, and so they are very popular in the common use. But what was at the beginning of their biggest advantage now became its biggest drawbacks, because the plastic pollutants actually released into the water do not stay in the place when they are released, but they might easily go to the river, fresh waters, or sea waters. And what is more important, they easily can go into the food chain. So they might be a serious. They might be a serious threat to our human beings, because the plastic pollutants under the affecting by the UV's radiation, oxidation agents, winds, and many other factors may be easily fragmented into smaller particles called microplastics. They are up to 5 micrometers, or even nanoplastics. They are smaller than 1 micrometers, and these particles can easily cross the cell membrane, or they can also easily cross the blood brain barrier. And what is because the treat of microplastic is not fully known. However, the microplastic pollutants may act as a carrier for other pollutants like heavy metal ions, pesticides, bisphenols, and many others. So they can, as I said, they can be very strongly affect our health and our human beings, as well as ecosystems. And what is the problem with the microplastic analysis is actually to properly prepare the samples for microplastic analysis. The samples of microplastic are very complex, because they might be composed not only from the synthetic polymers but also from the natural polymers. They can also consist of numerous of other inorganic compounds. So to separate the synthetic polymers from the complicated matrix is a very serious challenge. And nowadays study on the development of preparation of the samples for microplastic analysis are being developed in many various places. This is one of the most commonly used technique for the microplastic sample preparation. As you can see, it takes up to 17 days, and it consists of numerous steps of repeated oxidation agent use, filtration steps, as well as enzymatic treatment, including use of proteases and cellulases. Such a complicated sample preparation is a cost effective. This is the very important. And what is more, use of so many filtration steps might lead to the loss of some plastic analysis, some plastic fibers, some synthetic polymers. Because we have a lot of various microplastic techniques for microplastic analysis, including visual observations as well as microscopic observation, going to FTIR analysis or micro-Raman analysis. We can also use, for instance, pyrolysis GC coupled with MS, and these techniques allowed us also to a quantitative analysis of the microplastic. Nevertheless, to properly analyze the microplastic, we need a proper samples that is ready to be analyzed. This is very important, because the first microplastic analysis in drinking water has been performed, is required in California They do this from 2021, and nowadays European Union is also close to force these requirements to microplastic analysis in drinking water. So the aim of our study was to develop multi-enzymatic biomembrane composed of enzymes from various catalytic groups for the use in synthetic membrane reactors for one-step sample purification prior to microplastic analysis. I would like to highlight that this is just a very, very general view of our idea. Because on the surface of the ultrafiltration or nanofiltration membrane, we would like to immobilize different enzymes of lipases, proteases, lactases, and cellulases, because these enzymes are capable to degrade, to convert actually the inhibitors that affect microplastic analysis. The idea here, as you can see, is the enzyme co-immobilization, and after that covering of this membrane by the polydopamine layer. But we would like to also produce the layer by layer membranes, as well as we would like to modify it at first the nanofiltration or ultrafiltration membranes to efficiently immobilize the enzymes. And today I would like to present the results only on the cellulases immobilization on the gold and aluminum made ultrafiltration membranes. And why only these two types of membranes? Because the membranes made from aluminum or from gold can be easily analyzed using LDIR equipment from Agilent. So this equipment is capable to analyze these membranes, the polymers actually based on these membranes, because these membranes actually do not give the signals in this analysis. So here you can see the morphological changes of the ultrafiltration gold and alumina and gold membranes. After immobilization and after at first glutaraldehyde modification of the membranes, and after that cellulase immobilization. You can see that pure membranes can be seen are characterized by quite flat structure with easy observed pores. Whereas after enzyme immobilization, some enzyme aggregates can be observed on the surface of the membrane, and this aggregates, there is much more of this aggregates after glutaraldehyde modification at first. And this is the result that enzyme has been efficiently immobilized onto the surface of the membrane. We have also used the optical microscopy to characterize the membrane after cellulase immobilization. And you can see, due to the magnification, aggregates are not so easily observed. However, the changes in the morphology of the membrane can be observed, particularly after at first modification by glutaraldehyde and after that enzyme deposition. We have also performed the FTIR analysis of the pristine gold and alumina and gold membranes, as well as after enzyme immobilization, and signals around 1000 and around 1650 centimeters up to -1 are characteristic for the enzyme structure and confirm that enzyme has been immobilized onto the surface of the membrane. Whereas in the FTIR spectra of the pristine membrane, there is no such a signals. We have also performed the AFM analysis to determine the changes in the structural properties of the membrane. Pristine membranes are quite flat and pores can be easily observed, whereas both after enzyme immobilization and glutaraldehyde modification and enzyme modification changes can be easily observed, and some aggregates can be seen that might be assigned to the presence of enzyme aggregates on the surface of the membrane. We have also determined, based on the AFM measurements, we have also determined the surface area of this membrane, and it can be seen that after immobilization as well as modification and cellulase immobilization, the surface area decreased. as well as stiffness of the membrane has been significantly increased after enzyme immobilization. Also, changes in the adhesion energy and adhesion force can be clearly seen. We have also used the water contact angle measurements to determine the changes in the hydrophilicity of the surface of the membrane. The pure ultrafiltration membranes are characterized by limited hydrophobicity, whereas after enzyme mobilization and immobilization and modification, improvement of hydrophilicity is observed because the drop is smaller and is better absorbed by the surface of the membrane. And from the practical point of view, this is one of the most important parameters because we have also determined the water permeability of the membrane before and after the enzyme deposition. And you can see that significant value of water permeability for pristine membranes can be observed around 6,000 liters per square meters per hour per bar, whereas after enzyme immobilization, the drop is easily observed, as well as after modification and immobilization. This is due to the fact that surface as well as pores of the membrane are covered by the both modifier agent as well as by the enzyme. And I fully agree with you that these are very preliminary study, and no results on enzyme activity, on changes in enzyme properties here are presented. By this project has been just started, and we are still going through the immobilization of other enzymes, as well as we will going to deeply characterize the obtained membranes. Nevertheless, in conclusions, the process of sample preparation for microplastic analysis is time consuming and requires a numerous of steps. So we need to shorten this process and reduce its cost. And sample preparation might be shortened due to use of biocatalytic membranes in where the catalytic conversion occurred of undesired compounds supported by the membrane filtration, and efficient membrane functionalization and cellulose immobilization has been confirmed in our study. Nevertheless, modification and immobilization strongly affect the properties of our tested membrane as well as their water permeability. As I have mentioned earlier, study in single and co-immobilization of other enzymes, including lipases, lactases, and proteases, are ongoing right now. And we are also going to product layer by layer catalytic membranes, as well as what is the crucial here, we have to determine the activity and catalytic properties of the produced membranes. Thank you very much for your attention.